Comparative anatomy is the study of similarities and differences in the anatomy of different species. The purpose of this dissection was to observe the anatomy structure of rats, which is similar to humans. Now for some background information. Humans and rats are both considered mammals. Some characteristics of mammals are that they are warm-blooded, they have vertebrae, they typically have hair or fur, and they usually reproduce through live births. Rats are medium-sized mammals which are also considered rodents. Humans are also called Homo sapiens. Their evolved body shapes help them survive in hot and cold climates. Some things that define them are their advanced ability to think, languages, religion, etc. Now in this video, we will discuss the anatomy of a dissected rat, including body systems like the integumentary system, the respiratory system, the circulatory system, the digestive system, the urogenital system, and some additional observations, all of which will be compared to the anatomy of humans. Now to start off, we're going to be talking about the circulatory system, which involves the heart, veins, and arteries of the rat. Veins, which are the blue tubes, are used to carry deoxygenated blood back to the heart and lungs. Arteries, which are the red tubes, carry oxygenated blood to the muscles and organs that need it. The heart is covered in a thin, tough membrane called the pericardium. Here the heart was removed from the pericardial sac. You can see the aorta, which is the heart's pumping chamber, the superior vena cava, which is the large vein that receives blood from the upper body, and the pulmonary artery, which carries blood to the heart, labeled on the screen. These structures all work together to control the movement of blood into the heart. Now here we have the inside of the heart, where the two atria and the two ventricles, each on the right and left side of the heart, can be seen. This is where blood travels inside of the heart. Blood is pumped into the right atrium and down to the right ventricle. From there, it goes to the lungs to pick up oxygen and is deposited back into the left atrium. Then it is pumped down to the left ventricle before the aorta pumps it back into the rest of the body. Now, when compared to humans, the rat heart is obviously smaller. However, they both have 4 chambers, the structure is basically the same, and they both pump blood in the same direction.
After cutting through the thoracic cavity, we located the trachea, which is a tube that extends from the neck to the chest. The larynx, the voice box which contains the vocal cords. The glottis, the opening of the trachea. The bronchi, where the trachea splits in the chest cavity. The bronchioles, which are located within the lungs, and the 2 small tubes that the bronchi split into. The lungs, which lie on either side of the heart. And the diaphragm, which lies just above the liver and is responsible for drawing air into the chest cavity. The function of the respiratory system in rats and humans are the same thing. They both use this system for the exchange of gas. The major difference between rats and humans is that in humans, the right lungs has 3 lobes and the left lungs has 2. And in rats, the right lung has 4 lobes and the left has 1.
This is the urogenital system. This means that it encompasses both the reproductive and the urinary tract, both together. The first 3 organs we see are the prostate gland, kidney, and seminal vesicle. The prostate gland and seminal vesicles are primarily used for secreting semen. The kidney is mainly used in regulating blood. The medulla and cortex are part of the kidney, and that's where blood passes through. The adrenal gland is used to secrete adrenaline. The next 2 organs are the vas deferens, which moves sperm or semen to the urethra, and the scrotal sac, which is like a housing for the testicle. Lastly, the epididymis is used to store the sperm that is sent from the testicle. In the cranial region of the rat, you have the incensors, vibrissae, nictitating membrane, eyelid, and pupil. The incensors are the teeth for the rat. The vibrissae are basically its whiskers, which help with scenting, and its pupils help with eyesight, and they have similar eyelids to humans. Next is the pectoral region, which has its front legs, the thoracic region, where the rib cage is located, and the heart, the abdominal region, where most of its digestional and organs are located, and lastly, the pelvic region, where its tail is located and its hind legs. The anus is also located in the pectoral region.
For additional observations, we removed the skin from one of the hind legs and observed the muscles. We found the tibia, which is a weight-bearing bone located on the medial side of the leg. Then we found the fibula, which is a thin bone of the lateral leg. Lastly, we found the patella, which is also known as the kneecap. Rats and humans have the same leg bones, but one difference is that humans have 2 legs and rats have 4 legs.
The digestive system of rats, like that of humans, works to break down food into energy and nutrients through chemical and mechanical digestion. The process of digestion starts in the mouth. Food is broken down through chewing and it enters the esophagus. There, it travels down the esophagus and into the stomach. Meanwhile, the pancreas produces digestive juices to break down glucose, carbs, and proteins. The liver produces juices called bile, which is digestive fat. This allows the bile to store fat. In the stomach, which is a sac-like structure, food is broken down into small particles. The food leaves the stomach and enters the small intestine, which breaks down the food even further by using the enzymes released by the pancreas and liver. What is left enters the large intestine where it is compacted. The cecum is a pouch that rats have to store excess bile and food particles before it enters the rectum. The rectum sends messages to the brain telling it that the rat is ready to release its waste. The waste leaves the rat through the anus. A rat's digestive system compares to that of humans in 2 major ways. First, the rat does not have a gallbladder because they don't ingest fatty foods, which would make the gallbladder useless. Additionally, they have an enlarged large intestine called the cecum, which breaks down grains and seeds by using the bacteria inside the cecum, which breaks down cellulose. They are similar in the sense that they are made up of the mouth, esophagus, stomach, small and large intestine, rectum, and anus. Additionally, in both rats and humans, the digestive system plays the pivotal role of turning the food we ingest into the energy and nutrients that we need. All in all, rats are used for comparative dissections because they are relatively similar to humans. This is due to the fact that they have almost identical genes. Millions of years ago, they evolved from the same common ancestor. Since they have very similar sets of DNA, rat DNA is used in labs to mimic the effects of drugs and diseases would have on a human body. Rats and humans, although not identical, have the same body systems and they perform the same functions. This allows us to study organ systems and study ways that we can alter and fix any diseases that may be present in humans. The digestive system of rats, like that of humans, works to break down food into energy and nutrients.